Hi class, and welcome to your next video on organic compounds. In this video we're going to talk about carbon, and we're going to talk about carbohydrates and lipids. So carbon is one of those compounds that is present in every living thing. It is an organic compound. Carbon-based molecules are called organic compounds. Carbon has 4 valence electrons, and for those of you who do not know, valence electrons are these outermost electrons. So having 4 valence electrons means that carbon can bond up to 4 times. So there are 8 spots on that outermost shell for carbon, and those other spots on that shell mean it needs to be filled, and so it can bond up to 4 times. So we see this in methane. Methane, carbon bonds 4 times with hydrogen. So its formula is CH4. Methane is the most simple organic compound, and hydrogen fills blank valence electron spots in its outermost shell. So we have a lot of organic variability with these organic compounds. So methane and other compounds that are composed of only carbon and hydrogen are called hydrocarbons. They can be various lengths, they can be branched or unbranched, they can be double bonds, triple bonds, or they can be rings. All of these are going to contribute to their organic variability, and we're going to take a look at how these change these compounds. So if we look at this here, we have all carbons and hydrogens in both of these compounds, ethane and propane. But if you look, ethane only has 2 carbons and propane has 3. So here our carbon skeletons are varying in length. They're both still hydrocarbons, but they're going to vary in length here. Next, we can vary in branching. So when we start out here, we have butane, and butane has 4 carbons. And we still have 4 carbons when we look at isobutane, but isobutane is branched here. And we have 3 carbons in a straight chain, and then we have a carbon branched off of a carbon. So we're branching these carbons. Next, we have a double bond, and that double bond can change places. So the skeletons may have double bonds, which may change that location. And 1-butene, we have a double bond at the first carbon. And in 2-butene, we have a double bond at the second carbon. So that location is changing. Next, we're going to talk about what actually makes those macromolecules. Those macromolecules are very, very important. But what is a macromolecule? Well, a macromolecule is just simply a large molecule. Macro means large. So they're polymers made from building blocks. And those building blocks are called monomers. So poly means many, macro means large, and mono means one. So monomers make up polymers, and polymers are macromolecules. So kind of get all of those words straight in your vocabulary. So these are like Legos or Lincoln Logs. A cabin, a house, your finished product is the polymer. You put all those pieces together, and now you have your finished product. That's the polymer. But you've used all those individual Legos or logs, which are your monomers. You've used those to build your finished product. So we use all those individual pieces, those monomers, to make our polymer. So how are polymers made? Well, we actually have a reaction in which we make our polymers. So we actually use what is called a dehydration reaction. It is also known as dehydration synthesis, and this links together monomers by removing water. If you see here, there's a hydrogen and a hydrogen on one end and a hydroxide on the other end of a monomer. And what happens is those form together to form water, and then they bind the monomers. So what happens in the end is you started out with 2 monomers, now you're going to have a longer polymer. So we take out that water and now we bind it and we have a long polymer. So how are polymers broken down? Polymers are broken apart by the process of hydrolysis. This is exactly the opposite of that dehydration synthesis, taking out the water. Now we're going to add that water back in and we're going to break apart the molecules. So here we have a long polymer and we want to break apart those monomers and the polymers, so we're going to add in water. And when we add in water, we're going to get our hydroxide and our hydrogen added into our monomer and polymer into the spots that they originally held. So all of these biological reactions, both the dehydration reaction and the hydrolysis are of this sort are mediated by enzymes, which are going to speed up these reactions in cells. And we're going to talk more about enzymes when we talk about proteins, and proteins will be on our next video. So let's go into a little bit about carbohydrates. You might eat carbohydrates. You might think of them like pasta. You might think about them like fruit, vegetables. But let's think about just what is truly a carbohydrate. Well, a carbohydrate is a monomer. Its monomer is made of what we call monosaccharide. And if it has 2 monosaccharides, it's called a disaccharide. And many of those monosaccharides together are a polysaccharide. Here I have a picture of glucose. You've probably heard of glucose. And fructose. You probably have heard of fructose in the form of high fructose corn syrup. So you've probably actually ingested a lot of these two. The 2 of these together make sucrose, which is actually table sugar. So let's look at 4 important examples of our carbohydrates. So starch is used for storage in plants, and this is an example of our starch. Remember, we're talking about everything is made of monomers. Well, we can see here all of these little glucoses here are linked together to make our starch, and we're storing that in plants. So if you've ever heard someone say that like a potato has a lot of starch, they're right, because that is where it is stored. It is stored in plants. Glycogen is for storage in animals. So you store glycogen in your body. Glycogen is stored in the muscles and the liver of animals. And this is what we use for energy before we use fat. And what we do is it's pretty much the same thing, but it's a chain, and then we branch off here. So as you can see, it's a branched polysaccharide, and we again use those monomers and we branch them together. Then we have cellulose. I have a picture of what cellulose looks like under the microscope, and then here is just a small portion of the cellulose structure. Now, cellulose is used for structure in plant cell walls, but cellulose is also something else. You know it as fiber, and humans cannot digest fiber. So it's that roughage that we talk about that we need in our diets. We have chitin, which is the structure of animal exoskeletons, and chitin is actually found in mushrooms. It's found in a lot of fungus as well. So let's move on to lipids. Lipids are hydrophobic, and they cannot mix in water. The oil and water thing is true of all lipids. It is water-fearing. Hydrophobic means water-fearing. It's used for energy storage and can contain twice the amount of energy as carbohydrates and proteins. However, this is not just used for energy. This has some other uses as well. We use lipids in many forms. So let's go through the forms of lipids. So what makes a lipid? There is no true monomer for a lipid, so make sure you know that. We have parts that make up lipids. There's no true monomer for a lipid. We have triglycerides, which are called fats, and that's what you ingest. Triglycerides are the thing that you ingest. And they are composed of one glycerol molecule and 3 fatty acid chains. That fatty acid chain can be saturated or unsaturated, and that's based on their bonding. So we know that saturated fats are bad for us. Well, they contain no double bonds, and they are usually from animal fats, and they're solid at room temperature. If they have one or more double bonds, they're unsaturated, and they actually come from plants generally. And even though you might find them in like nuts, their oils, which is actually where the lipid comes from, is actually liquid at room temperature. Next we have phospholipid, which makes up the cell membrane, and it's actually a bilayer on the cell membrane. So we have 2 layers of these phospholipids going all the way around the cell. And this protects our cell because remember we talked about them being hydrophobic. Well, they aren't going to let things in unless they're polar, and certain things can't get in and can't get out. And this is really going to protect our cell. Now we do have other mechanisms, but we're going to talk about that a little later. They contain 1 phosphate head, so this polar head and phosphate group you see, and then they contain 2 fatty acids, and one is saturated and one is unsaturated. And this is actually the structure, this is the simple structure of a phospholipid. Then we have steroids. Steroids are fairly simple. They have a ring-like structure. And we have 2 examples, a cholesterol and a sex hormone. So just to review, we went through hydrocarbons, remember methane. We went through the organic variability. We have various lengths, branching and unbranching, double chains, or double bonds and rings. We went through macromolecules, and monomers make up polymers. We went through dehydration reaction, and we went through hydrolysis. We went through carbohydrates and the fact that their monomer is a monosaccharide, their polymer is a polysaccharide, and we have a special name for 2 monosaccharides together, which is a disaccharide. And we went through lipids, and they cannot mix in water. They are hydrophobic. They have no monomer. We have triglycerides, phospholipids, and steroids. Okay, I hope you got great notes, got lots of the figures down, and we will talk about this later in class.